Now that we have talked about the general characteristics of the innate immunity, let's look at the previously mentioned mechanism that executes its functions in detail. Starting with phagocytosis. One of the first features of the immune defense system that a foreign organism encounters after being introduced through a cut in the skin is the phagocytic white blood cells, leukocytes, such as macrophages, which congregate within minutes and begin to attack the invading foreign microbes. Phagocytosis is an important mechanism. This mechanism of phagocytosis consists of a series of events including attachment, endocytosis or ingestion, and digestion of microbe. Starting with the first step, that is attachment. Following chemotactic signals, phagocytes like neutrophils reach at the site of infection. Similarly, other phagocytes are also guided by chemotaxis, and that chemotractant are usually inflammatory cytokines or microbial products. So following chemotaxis, once phagocytes reach to the site of infection, now they are in close proximity with microbe. Here the process of phagocytosis activates, and this activation is triggered by binding through one of the receptors present on its surface, known as pattern recognition receptors. In general, PRs on phagocytes can detect pathogen-associated molecular pattern. These are actually a diverse set of microbial molecules that share a number of different general patterns or structures that alert immune cells to destroy intruding pathogens. Normally, the most common PMPs are usually mannose-binding lectin that are the complex carbohydrates found in the cell wall of yeast, and lipopolysaccharides, LPS, that is cell wall component of gram-negative bacteria. This mechanism of attachment between PRRs and PAMPs is common for non-capsulated pathogens, but the capsulated ones can't be detected by PRRs. So for their attachment with phagocytes, they need another additional step. That is called opsonization. Well, opsonization is the important process in your defense system by which particles or complexes are made readily ingestible for uptake by phagocytic cells. In simpler words, specific serum proteins, known as an opsonin, coat the invaded bacteria. This opsonization allows the phagocytic cells, such as macrophages, to pick up the pathogen more easily and faster, and engulf them by the process of phagocytosis. Here, the most essential opsonin of all is complement C3b, which is a particularly important opsonization protein for controlling bacterial infections by this mechanism. Now, opsonization allows killing of gram-positive bacteria, like Staphylococcus species, that are resistant to killing by membrane attack complex. After attachment, next step in phagocytosis is endocytosis or ingestion. Endocytosis is the process to take in substances from outside the cell. Same is needed for phagocytosis now. When the foreign particle or pathogen gets attached at the receptors of phagocytes, it is then pinched inside of the cell, together with a part of phagocytic cell membrane. This vacuole that is composed of phagocytic cell membrane with a captured pathogen inside is called phagosome. Phagocytic cells contain intracellular compartments or vesicles, one important vesicle that contain antimicrobial substances inside are lysosomes. So once the particle is internalized inside the phagosome, this vacuole can fuse with lysosomes to form another vacuole called phagolysosome. This phagolysosome is a microbicidal vacuole, and it contains many substances that have ability to kill microbe. This formation of phagolysosome is also called as vacuole formation. Within these microbicidal vacuoles, invading particles are further processed. Now we move towards final step of phagocytosis, that is killing or digestion of microbes. A lysosome or phagolysosome is able to break down the stuff inside of itself by 3 mechanisms, including low pH, by hydrolytic enzymes and other microbicidal substances or scavenger molecules. Firstly, it drastically lowers the pH of its internal environment. Lowering the pH makes the environment inside the phagolysosome makes it very acidic. This is an effective way of killing or neutralizing whatever is inside the phagolysosome, so it cannot infect other cells. While other microbicidal components of the phagolysosome are scavenger molecules, such as lactoferrin that sequesters the iron required by some bacteria. So all these things like acidic medium or low pH and digestive enzymes cause killing or digestion of trapped microbe, leaving behind a sac filled with indigestible material, called as residual body. So once the contents have been neutralized, the phagolysosome forms a residual body that contains the waste products from the phagolysosome. The residual body then travels towards the boundary of the cell and is eventually discharged from the cell by a process called exocytosis. Here, phagocytes can also undergo partial digestion of microbial protein that can be displayed on the surface of the APC associated with an antigen-presenting molecule, such as MHC class I or MHC class I, for recognition by T and B lymphocytes. Such process is called antigen presentation. That's how macrophages play dual role as phagocyte in innate immunity and as antigen-presenting cell in adaptive immunity. Such process is called antigen presentation. That's how macrophages play dual role as phagocyte in innate immunity and as antigen-presenting cell in adaptive immunity. But neutrophils normally don't find time to act as APC as their lifespan is short. The 2 major phagocytes are macrophages and neutrophils, and the main mechanism of phagocytosis and microbial killing is same in both these cells. So this was all about detailed mechanism of phagocytosis. One of the immune response generated by immune system against that invader is inflammation. Although sometimes it becomes hard to characterize it under good immune response, as it can cause even more damage sometimes. But still, inflammation is very important to immune response. Let's look at its definition. Inflammation is a nonspecific host defense mechanism that combats infection by all the means at its disposal. Other than infection, it also responds to any type of tissue damage or injury and resolve or repairs it. Here, the type of white cells involved are neutrophils, macrophages, eosinophils, or types of lymphocytes, like T or B lymphocytes, vary with each type of inflammation. So we can say, inflammation is a normal response of the body to protect tissues from infection, injury, or disease. Number of stimuli can lead to inflammation, such as burns, physical injury, toxins or trauma, disease, or a medical condition. But most inflammation is a response to the infection by any pathogen or microbe. So these stimuli, and thus the intensity of inflammatory responses, may vary. But the main goal of inflammation is to restore balance. Inflammation is divided into 2 types, acute inflammation and the chronic inflammation. Well, acute inflammation typically lasts only a few days, but its onset is rapid. While chronic inflammation typically lasts weeks, months, or years, or even indefinite period. And it has slow or delayed onset. In case of acute inflammation, on the release of inflammatory mediators, inflammatory cells mobilize to the area of infection or injury, and by certain mechanisms, resolve the problem or heal the tissue. Whereas chronic inflammation causes continuous tissue damage that can lead to organs failure. With respect to the immune response, another difference is that, During acute inflammation, innate immune cells form the first line of immune defense and regulate activation of adaptive immune responses. By contrast, during chronic inflammation, these roles can be reversed. Adaptive immune responses can cause ongoing and excessive activation of innate immune cells. Mechanism of acute inflammation, which supports the innate immune response, is dependent on inflammatory mediators, also known as cytokines. When pathogens breach the external protective barriers, the initial reaction to those pathogens is usually an acute inflammatory response, involving an influx of leukocytes, complement previous antibodies, if any, and other plasma proteins into a site of infection or injury. And these influxes are triggered by inflammatory mediators. Inflammatory mediators are actually chemical signals that generates inflammatory response. Here, a complex variety of mediators are involved in acute inflammatory responses. Some act directly on the smooth muscle wall surrounding the arterioles to alter blood flow. Others act on the venules to cause contraction of the endothelial cells, an important step in inflammation. The migration of leukocytes from the bloodstream is also facilitated by soluble mediators. Other mediators act as chemotactic factors to attract the leukocytes to the site of inflammation by a process called chemotaxis. So by one way or other, all these mediators initiate inflammatory response. These mediators include histamine, tissue necrosis factor, bradykinin, interleukins, leukotrienes, platelet activating factor, etc. After release of inflammatory mediators, the leukocytes that are charging along in the blood vessels need to be redirected to migrate into the inflamed site. Several classes of substances or chemicals subserve this role as well. So once leukocytes are migrated towards the site of inflammation, then there starts the initiation of the acute inflammatory response. Locally, the acute inflammatory response include capillary vessel dilation or vasodilation, exudation of plasma proteins that leads to edema, leukocyte adherence to endothelium by the help of various chemicals and receptors, chemotraction and local activation of leukocytes. Together with triggering of these inflammatory responses, chemical mediators also initiates acute phase response that is characterized by promotion of marrow production of neutrophils and monocytes. Also during this phase, these macrophage-derived cytokines activates Th1 and NK cells that secretes interferon gamma, IFF&G, and more tissue necrosis factor, TNF. This interferon gamma has a direct effect on the hypothalamus and increase the body temperature and can cause fever, which impairs pathogen reproduction. In acute phase response, liver is also stimulated to produce complement and other proteins, mainly C-reactive protein, CRP. Here, CRP is an important protein produced in the liver that binds to phospholipids on the surface of bacteria. CRP then acts as an opsonin stimulating phagocytosis. CRP also activates the complement system and acts as a reliable marker for the diagnosis and monitoring of inflammatory responses. Often, systemic features of an acute phase response include fever, high white blood cell count, loss of appetite, and malaise. At its most extreme, an acute phase response may lead to septic shock, other than activation of acute phase response. So this was an overview of all inflammatory responses. The acute inflammation initiates two types of events, namely the vascular events and the cellular events. You can check out our video on acute inflammation and chronic inflammation in the immunology category to get an in-depth look into both these topics. Moving on, we have mentioned complement proteins and complement system several times. Let's into it in some detail. The complement system is a part of the innate immune response, but we see that the complement system acts as a bridge between the innate and acquired immune response, as one of the pathways of activation is antibody-dependent. The complement system is composed of about more than 20 different proteins that work together to destroy invaders and to signal other immune system players that the attack is on. It is called a complement because it complements the ability of phagocytic cells and antibodies. The complementary proteins are produced by the liver, which circulate in the blood as enzymes called zymogens. They normally live in an inactive state until the cascade events start and activate the constituents of the complementary system. Other than infected cell lysis and immune clearance, the function of the complementary system is to cause inflammation so that more immune cells can join the battle against a pathogen, and opsonization of pathogens can occur. While the complement proteins assist in pathogen-killing VIA3 pathways, the classical complement pathway, the lectin pathway, and the alternative complement pathway. The classical pathway is antibody-dependent and thus part of the acquired immune response. Lectin and alternative pathways are part of the innate system as they can be activated upon first exposure to a pathogen. While these all three pathways follow the same pattern, once the C3 protein is activated. In other words, the pathways differ from one another in the way of their initial activation step, while the rest of the pathway is the same. As you can see, the protein's fragments that are produced after the activation of complement cascade have a number of functions such as upregulating the inflammation and opsonization process. We already had a detailed discussion on these three pathways in our video on the complement system. So here we are just going to skim through them for revision. Starting off with the classical pathway. So the first steps of the classical complement pathway require the binding of antibodies to the surface of the target bacterium. The antibodies then become targets for one particular complement protein complex, known as C1. C1 binds to the tail, known as Fc region, of the antibody. Once bound, C1 initiates a cascade of cleavage and reforming of complement complexes that ends in the binding of several complement proteins to the surface of the bacterium in the form of a membrane attack complex, or MAC, or can generate opsonins that label a bacterium for destruction. Then membrane attack complex, or MAC, can insert into the cell membrane of pathogens. There it produces pores that allow the entry of membrane-damaging molecules, such as lysozyme, and makes the bacterium susceptible to osmotic lysis. Now the second pathway, the alternative complement pathway, does not require antibody to initiate the lysis of bacteria. Here, complement proteins from a complex known as C3 directly bind to bacteria and activate downstream components in the complement cascade, once again ending in formation of membrane attack complex. That causes lysis of the bacterium, just like the classical pathway. Now, moving on to the lectin pathway. During the lectin pathway, mannan-binding lectin, or MBL, binds to proteins containing mannose residues that are found in some types of bacteria, such as Salmonella species. Once bound, MBL forms a complex with an enzyme called MBL-activated serine protease, or MAASP. In this form, this enzyme activates C3 convertase by cleaving C2 and C4 complement components that participates in forming membrane attack complex, and the rest of the pathway is same. That is all about the innate immune system and the constituents that play their part in order to fight off invading pathogens.
Explore our extensive library of over 1,800 video lectures to learn about a wide range of topics. Only on sqadia.com.